Hello everyone. Today we are going to talk about transport, specifically gated transport into and out of the nucleus. This is section 1 on our 6-section series on transport. So in our textbook, the transport of molecules between the nucleus and the cytosol is covered briefly on pages 972 and 973. So we have some of the important biochemistry discussed here, but I wanted to put this into more of a microbiology context. So that's the purpose of our talk today. So that's what we will do. So this is my outline. So I have 6 sections. So today, this is section 1, transport into and out of the nucleus. So we start first by talking about evolution of organelles because we want to focus on the nucleus. So imagine we have genomic DNA of an archaeon, so that's shown in this kind of like this little sort of red scribbly stuff right here. And then the cell wall is lost. So that facilitates horizontal gene transfers, which could increase genetic diversity. And then now that you don't have your cell wall anymore, you can undergo phagocytosis and digestion of other prokaryotic cells, and that sort of speeds up the evolutionary process. But the next step here is that the membranes will enclose the chromosome to protect it, because if this is our genetic material that will specify all the characteristics of the organism, we need to protect it. So we have this a membrane that that encloses and invaginates around this around what will become the nucleus. So this forms a membrane around the nucleus, but it still allows access of the DNA to the cytosol. So you can see that there are these little gates right here where we have access. We have transport between the nucleus and the cytosol, which we will need to do. So if we continue in our evolutionary discussion right here, if the cell has engulfed a pro-mitochondrion right here, then the mitochondria then become the powerhouses of the cell and are able to use oxygen in order to extract as much energy from glucose as possible. So the first eukaryotic cells are in fact aerobic. So these organelles, though, as you can see, if they've been engulfed in this way, then they are isolated from this sort of this cellular trafficking right here. So here we can see our enclosed nucleus and these membranes, and then these membranes right here become part of the endoplasmic reticulum that is contiguous with the nuclear membrane. So this evolutionary scheme is used to group compartments into 4 families. So we have the nucleus and the cytosol. So we have pores through the nucleus. They are topologically continuous and functionally distinct. That allows material to be transported between the nucleus and the cytosol. We also have organelles that function in the secretory and endocytic pathways. So this is part of cellular trafficking. So that would be the endoplasmic reticulum, Golgi, endosomes, lysosomes, transport vesicles, and peroxisomes. They're all grouped together. The mitochondria, as we saw, are isolated. So we'll talk about those. Plastids are similar, but plants are beyond the scope of this course. So I start out this way because I want to show you what the topologically equivalent spaces are. So if we have the inside of the cell colored in gray in this picture and the outside in red, what that means is that if you are inside, say, the Golgi or a cargo molecule vesicle right here, which then fuses with the membrane to release its cargo, the outside of the cell and the inside of these organelles are topologically equivalent. And we can see that in the whole cell over here too. So here's the nucleus and the endoplasmic reticulum, and then this is the Golgi apparatus and the lysosomes, and we have endosomes and secretory vesicles. Notice that they're all colored inside with red. So the inside of those organelles is topologically equivalent with the outside of the cell. we should think about that in terms of membranes too. So if you think about the outer leaflet of the membrane and how it faces the outside of the cell, that's going to be the inner leaflet here, because the same leaflet of the membrane that faces the outside will face the inside of these organelles, and then the same portion of the, here it would be the inner leaflet that faces the cytosol. That's going to be the same as the outer leaflet of these organelles. So the side that faces the cytosol, whether you're in the plasma membrane or one of these organelle membranes, is going to be the same. So I uploaded to Sakai a Word document where I pasted this figure. And if we were in class, what I would do is I would photocopy for you this for you in color, and I would give it to you. And when we went through any of these sections, I would say, get this out and make sure you can remember where we are. We have different types of transport: gated transport, transmembrane transport, and vesicular transport. So we'll talk about all of these in turn. Today, we want to talk about gated transport, and that's shown in red in this picture right here. And the arrows are going in both directions. So what that means is that we can transfer material from the nucleus to the cytosol and from the cytosol to the nucleus. So the authors of this book redraw their picture, and they just focus on what we're talking about. So we're talking about moving between the cytosol and the nucleus, and in both directions. So transport in and out of the nucleus. If we want to go in to the nucleus, the kinds of proteins that need to go in, remember, protein synthesis occurs on the ribosomes in the cytosol. So once you make those proteins, then they have to get into the nucleus. So histones that bind to the DNA, transcription factors, DNA polymerase, RNA polymerase, all of those proteins that we talked about that are involved in DNA replication and transcription, they all have to get into the nucleus. So their import is dependent on a GTPase called RAN. How about transport out? What goes out? There's RAN-dependent export. There's also RAN-independent export. We already alluded to this a little bit in chapter 26 when we talked about how messenger RNA and ribosomes are exported out of the nucleus. So that occurs via a RAN-independent process. So we need to talk about RAN, so we'll talk about that today. But first, we want to talk about these the nucleus and how it's arranged and how we visualize this in context with that evolutionary scheme that I gave you. So this is sort of a cutaway version of the nucleus, and we have an inner nuclear membrane and an outer nuclear membrane, and where there are breaks in these membranes, those are the nuclear pores. Notice that this membrane is contiguous with the lumen, or the inside of the endoplasmic reticulum. This green stuff right here is meant to represent the meshwork of the nuclear lamina. Think of it as kind of like a chicken wire kind of structural protein that gives the nucleus its shape and its structure. So these spaces right here, these holes right here, and these spaces right here, those are called nuclear pore complexes, and they have to penetrate both membranes if you're going to get things in and out of the nucleus. So there's bidirectional traffic, and that has to be regulated because you want the proteins that need to get into the nucleus to get in, and not other things that are not supposed to be there. So I have a couple of diagrams right here. Again, here's another cutaway version right here. And to orient ourselves, the cytosolic surface is on the top, and the inside of the nucleus is on the bottom. So again, if we sort of have this cutaway version right here, there are these proteins that they're sort of called ring proteins because they sort of form this ring for this transporter. And then there are this sort of these a nuclear basket right here. And then these kind of like little spaghetti kinds of things right here, those are proteins that will be involved in allowing the passage of certain entities across the nucleus. So those are called FG nucleoporins. Out in the cytosol, we have these cytosolic fibrils, so their job is to kind of reach out and grab things that are supposed to get into the nucleus. So there are there are entities that can go past this passively, and there are other things that are much larger that need to move these things out of the way. So that would constitute active transport, and we would need hydrolysis of ATP in order to accomplish that. So here's another view from a different textbook. So again, to orient ourselves, we're inside the nucleus here, outside the nucleus here. So we have these cytoplasmic filaments, and then shown here in purple in this artist rendering are the FG nucleoporins. So those are rich in Phe and Gly, so that's why it's called FG, so F for Phe and G for Gly. So those are the FG nucleoporins, and then these in green are these structural proteins right here. There's the nuclear lamina right here, and this is the nuclear basket that faces the nucleus. So these nucleoporins form sort of a gel-like matrix. So there are a lot of proteins here, as you can see. So it allows diffusion of small molecules, whereas if you're larger than 40 kilodaltons, you can't get past unless you have some kind of, and here I use the word unchaperoned. So chaperones aren't always involved, but there must be some kind of way to kind of push these things out of the way in order for larger entities to get across. So what's interesting is you can do these experiments to determine what kinds of signals proteins possess that allow them to get inside the nucleus. So these are nuclear localization signals, so we'll just call them NLSs for short. So this is a T antigen, and it has its normal nuclear import signal, which you can see is rich in basic amino acid residues. And in this picture, you can't see this, but these are just nuclei. They're not cells. Like the boundaries of the cells, you can't see. It's too dark. But the T antigen with its nuclear localization signal is attached to some kind of fluorescent label so that we can see it. But suppose you do some site-directed mutagenesis experiment and you change one of those lysines to a threonine. And what happens is that is enough of a mutation in this nuclear localization signal that the pore doesn't recognize it and the protein can't get inside. So if these are the nuclei right here, you can see now all of the fluorescence is in the cytosol. So if you mutate the nuclear localization signal on the protein, then you can't get inside. You can also maybe have a little fun with this and see if you can get things in the nucleus that aren't supposed to be here. So pyruvate kinase, you might remember, catalyzes the last step in glycolysis, and it's supposed to be in the cytosol because that's where glycolysis takes place. And accordingly, pyruvate kinase doesn't have a nuclear localization signal, but you can fuse one. You can take one of those nuclear localization signals and again do some genetic engineering and fuse it to the pyruvate kinase gene so it expresses a protein with a nuclear localization signal, and you can see if you look in part B, it does end up in the nucleus. So these kinds of experiments can help you determine exactly what amino acids are necessary to allow the localization of a protein into the nucleus. So we have nuclear localization signals, NLSs. So proteins imported into the nucleus must have those. And the nuclear localization signal is recognized by a cytosolic protein called importin. So it has a name that sounds like what it does. Proteins that are exported from the nucleus have a nuclear export signal, or an NES. That's recognized by a nuclear protein called exportin. So there are proteins that shuttle back and forth, so they need to have both. So that means that the NLS can be recognized by important and then the NES by exportin. Okay, so how does this work? So I mentioned RAN. So how does RAN work? Well, there are some general principles here. First, RAN is a monomeric GTPase. So you've heard of GTPases before. So if you think about signaling pathways, glucagon signaling pathway that we talked about last semester, the receptor is associated with something called the G protein. That is a trimeric. Remember, it has an alpha, beta, and gamma subunits. That's a trimeric GTP protein. This is monomeric. So there are quite a lot of members of this family, and I will introduce you to them in the next few weeks, and this is one of them. But the other way these things work is they're associated with gaps and GEFs. Okay, so a gap, so here, this is called RAN gap. So a gap is a GTPase activating protein. So a GTPase activating protein does just like what it sounds like. It hydrolyzes GTP to GDP. So monomeric GTPases like RAN and other ones, they are inactive when they have GDP bound. They are active when they have GTP bound. So a gap will hydrolyze the GTP to GDP and inactivate RAN. We also have to go in the other direction. So here we have a GEF. So GEF is a guanine nucleotide exchange factor. So that doesn't carry out a reaction like the gap does. It's just a swap. So it swaps GDP for GTP and activates RAN. So what we have is, what's shown in here is we have inactive RAN because it has GDP bound to it. And if the inactive RAN goes through this nuclear pore from the cytosol to the nucleus, it will encounter RAN GEF. So RAN GEF will swap the GDP for GTP. So now the RAN GTP is active. And the cycle completes when the RAN GTP goes out of the nuclear pore, through the nuclear pore to the cytosol, and then we have RAN gap that will hydrolyze that GTP to GDP, and now RAN GDP is inactive. So notice that the way that this works is that these are physically separated. You have RAN gap in the cytosol and RAN GAF in the nucleus. Okay, I think I might have said this. Okay, so RAN GDP is in the cytosol because the RAN gap is cytosolic. So again, notice here that the RAN gap is cytosolic. So because you have these localization of these proteins the way you do, that indicates that you've got a flow in this direction because of the concentrations involved. So since we're going along a concentration gradient, they don't need to interact with those FG nucleoporins. Same thing over here. So RAN GTP is in the nucleus because the RAN GEF is nuclear. So again, it can go back into the nucleus along a concentration gradient. So interacting with the FG nucleoporins via active transport's not required here. Okay, so I have lots of pictures from lots of different textbooks. Okay, so let's talk about how import happens. So the last slide just outlined the principles. So these are the players up here at the top. So here's our RAN GDP, and remember that's inactive. Here's RAN GTP. So when GTP is bound, we have a conformational change that occurs. Maybe they should have made it a different color, but anyway, this is important. And this is, so this is just labeled as cargo. So this is a protein that has an NLS, or a nuclear localization signal attached to it. So this is a protein that is meant to be inside the nucleus. So this figure shows how that happens. So we're going to start here. Here is our protein, our cargo, with its nuclear localization signal, and it is going to bind to importin. So importin is located in the cytosol. So now it goes across, and so that this cargo complex goes across. So now my cargo, my protein, is inside, bound to importin. So the important interacts with these FG nucleoporins because now you've got this big complex. So in the last slide, I told you that RAN doesn't have to do that, but now that you're bound to this big complex, you do have to. So the importance job is the one to interact with the FG nuclear proteins and kind of push them aside so this large complex can get through. Okay, so now we're in the nucleus. So there I am. And what happens now is, remember we said that RAN-GEF was located in the nucleus. So the GEF is going to swap the GDP for GTP. So now this was RAN-GDP, which is inactive. Now that I have the swap, this is RAN-GTP, which is active. So there are conformational changes that occur, and one of the things that happens is that you release your cargo, and now the cargo will go off and do whatever it does. Maybe it's a histone protein, so you need to bind to the DNA. So now what happens is important now, its job is to get the RAN-GDP back. Okay, so the release of the cargo happens. So that is actually dependent on RAN-GTP. So if you can't convert the RAN-GDP to the GTP form, you can't release your cargo. So this part has to happen. Okay, so we have our important, okay, so we've released our cargo, it's going to go off and do whatever it does, but now we have to complete the cycle. So we have to get the important back into the cytosol. So the RAN-GTP will get it there. So we go across, and the important, once again, will interact with those FG proteins. And now here we are in the cytosol, but in the cytosol is RAN-GAP. So RAN-GAP will catalyze the swap of the GDP for GTP. And now we have RAN-GDP, which is inactive. And we're going to release the important, and now the important can carry out the cycle again. So there is one thing that I added that the artist didn't show. So here's my addition right here. There it is. Okay, so this only will work then if there's another way that the RAN-GDP can get back from the cytosol into the nucleus in order to complete this process. So the RAN-GDP is able to do that and go through this nuclear pore complex, and it doesn't have to interact with the FG nucleoporins in order for that to happen. So anyway, so I added that to the picture because this really does complete the entire cycle. All right, so what about export? What if you're trying to export cargo out of the you're trying to export cargo out of the nucleus and into the cytosol. So we're going in the opposite direction, nucleus to cytosol. Okay, so here are the players. Maybe they should have drawn some of these differently, because here's our RAN-GDP, so that's inactive. Here's RAN-GTP, which is active. So here's exportin. So like I said, maybe they should have drawn that in a different color, and maybe they should have done this cargo differently too. So this is cargo, that's a protein, and now this has a nuclear export signal right here. So this shows that this protein that is in the nucleus needs to get out into the cytosol. So here we are inside the nucleus, and here is our cargo. So now we have this huge cargo complex because we have our cargo with its nuclear export signal and this exportin protein that recognizes it. And then we also have to have RAN-GTP bound. So there's our cargo complex. So this is huge, so it again goes across, and it has to interact with these FG proteins and kind of shove them out of the way. So now here we are in the cytosol. So what happens now, we said, remember that RAN-GAP is in the cytosol, so we're going to hydrolyze the GTP to GDP, and then that causes the dissociation. So here's our protein, it's going to go off and do whatever it does, but we've released the exportin, and now the RAN-GTP is then converted to RAN-GDP, and now both the exportin and the RAN GDP have to come back into the nucleus to complete the cycle. And once the RAN GDP is there, the GEF then has to do the swap of the GDP to GTP to convert the RAN GDP back to RAN GTP in order to start the whole cycle over again. All right, so notice that the RAN GTP is part of the cargo complex for export but not import. Okay, so I've just shown you bidirectional transport between the nucleus and the cytosol. We do have some proteins that have both, in which case there's a shuttling of these proteins in and out of the nucleus. And in other cases, the process is regulated, and you can turn on or turn off these nuclear localization signals. So I have this one example right here that's really interesting. So this is a resting T cell in the immune system, and there is a protein called NFAT. So that's shown here in gray, and it's a gene regulatory protein. So gene regulatory proteins, we spent a lot of time talking about those. They bind to the DNA and either activate or inactivate the transcription of different genes. But in this case, we in the resting T cell, the gene regulatory protein is kept outside because we don't need it yet. So what happens is, in order to keep it inactive, it's phosphorylated. So when the T cell becomes activated, when we talk about cell signaling, we'll do that in the last week of school. We'll talk about how some signaling processes involve just a flooding of calcium ions from the endoplasmic reticulum into the cytosol, and that's a signal. So there's this protein right here shown in blue, and it's called calcineurin. And when calcineurin is bound to calcium, that activates it. So calcineurin is a phosphatase, and phosphatase will do what phosphatases do, and it will do a hydrolysis, and we'll get rid of these phosphates on the NFAT. So now that this thing is dephosphorylated, that involves huge conformational changes. So what happens here is a nuclear import signal is now exposed, and this would be a nuclear export signal that was blocked. So now it is time for this thing to go into the nucleus, and it's not shown in this figure, but this whole RAN thing that I showed you in the last couple of slides, that's how it happens. So we'll leave out that detail for now since I already went through it. Okay, so here we go. Inside the nucleus we get, and then now this is a gene regulatory protein, so it's able to bind to the DNA and do what it's supposed to do. So that's this signaling event that activates the T cell. So an activated T cell has to synthesize proteins that cause it to be activated. So when the signaling event is over, what happens is all of those calcium ions that flooded the cell from the ER get pumped back in. So now the calcium, the T cell reverts back to its resting form, and now the calcium concentration is low. So what that means then is that the NFAT dissociates from its phosphatase, and there's a kinase in the nucleus that phosphorylates it. That now is going to cause conformational change. It's going to hide its nuclear import signal and expose its nuclear export signal. And now that same kind of a process I just showed you before involving RAN will allow this thing to leave. So that means we're able to activate genes, certain genes to be transcribe certain proteins during the activation cycle of a T cell, but not when it's resting. Okay, so I think I already said that. Okay, so what's being shown right here is this: the NFAT is going back now into the cytosol. It's able to do that because it's got its export signal, and now it just hangs out in the cytosol of the cell until a signaling event happens once again to flood the cell with calcium, so it happens all over again. So those are all examples of import and export involving RAN, But we also want to talk a little bit about messenger RNA leaving the nucleus, and we kind of talked about a little bit about this before. So this happens, and this is independent of RAN. So we did talk about earlier that pre-messenger RNAs need to be spliced and polyadenylated. It needs the poly A tail, needs the cap, and all of the introns need to be spliced out. So the RNAs that are properly processed and are allowed to leave have to be recognized as such so that they are export ready, and then they're allowed to leave. So if they're not properly spliced or polyadenylated, then there's a complex of nucleases inside the nucleus. It's called the exosome that will degrade it. So there's a cap binding complex that binds to the cap on the messenger RNA, and that helps it protect it from attack by these exosomes. And it also allows a signal that says, yes, you're allowed to leave. And we also have the poly A binding proteins that bind to that poly A tail. That also prevents exosome attack, and that also is a signal for export. So this is going to allow the export-ready RNA to go through this nuclear complex and go from the nucleus to the cytosol where it can be translated into protein. So this shows a little bit more detail than we had talked about before. So you can, here we are in the nucleus, it's shaded in blue. So you can barely see this. So the messenger RNA is this red string right here that you can just barely see because it's just covered with proteins. There's the cap binding protein right here. We have the poly A binding proteins here. And there are, we didn't mention this before, but we have NXT1 and NXF1. So this is nuclear export transporter 1 and nuclear export factor 1. Now they don't involve RAN, but they do interact with those FG domains. So there are also these heterogeneous nuclear ribonuclear proteins, and they also have a role to play in ensuring that this messenger RNA is properly capped, properly spliced, and has its poly A tail, and then is allowed to leave. So it leaves through this the nuclear pore, and quite a lot of these proteins come back into the nucleus and are recycled, and then new proteins bind, such as the eukaryotic initiation factor 4E, because it says, okay, here is our cap, let's translate this thing, and let's recruit these initiation factors to make that happen. So, so this is a RAN independent mechanism. RAN is not involved here. So here's another picture that shows kind of some of the same things. So here's our messenger RNA. There's the cap. There's the poly A tail. Oh, sorry, I was starting in the wrong place. I should be starting down here at the bottom. This particular artist likes to start a lot of their pictures at the bottom, so sometimes I can get confused. Okay, and I don't mean to confuse you. Okay, so anyway, so here is the messenger RNA. There's the cap, there's the poly A tail, and then these are the factors that allow it to interact with these FG proteins to allow it to leave the nucleus and go out into the cytosol. So again, the job of the NXF1 and the NXT1 are to kind of shove those FG domains out of the way so they can leave. What's happening here, and this is discussed in our textbook, is this DBP5. That's a helicase, and it associates with the nuclear pore complex. And what it does is its job is to, once the messenger RNA has transported itself across this nuclear pore complex, it strips off the NXF1 and the NXT1, and then that helps pull it into the cytosol. And the way that happens is that it's called a helicase. It's sort of stripping off different kinds of molecules, not kind of the way we're thinking of helicases working in DNA replication, but ATP hydrolysis is required, just like it is for all helicases. So that ensures that this is a unidirectional process. So we strip off these proteins, they're allowed back in, but now our messenger RNA is in the cytosol, and the ribosomes can hop on, and we can start the translation process. So when the NXT1 and the NXF1 come back, it's not shown in this figure because that would make it too complicated, but they actually do use the RAN system to get back. So the messenger RNA can leave without using the RAN process, but the proteins that allow it to do that, when they come back, they use the RAN process. So export ready messenger RNA is allowed to leave through this nuclear pore. So we have these heterogeneous nuclear ribonuclear proteins, and their job is to distinguish the mature messenger RNA for processing debris. And again, the artist here has shown the different things that do that. The cap binding protein, the poly A binding proteins. We have this exon junction complex, and its job, along with the HNRNPs, is to ensure that the messenger RNA has been properly spliced. So if you went back to the splicing section in our notes, you'd find that the SR proteins and the EJC and the HNRNPs are all involved with that. Once we have assured the machinery that we're ready to go, then it is allowed to leave. Some of the proteins go along for the ride, as we showed before, and go back in, but some of them dissociate from the messenger RNA before as it's transported into the cytosol. And now here we are. And now the messenger RNA has its cap and its poly A tail. So here is in these eukaryotic initiation factors, one shown in brown and one shown in pink, and they bind to the cap. And then you form this circular complex, and now you can translate your messenger RNA. So this is what we've done today. We've talked about transport in and out of the nucleus. So here's our diagram, and this transport is called gated transport. And the arrow is shown in red. So red means gated transport. The arrow is going in both directions because we can transport entities from the nucleus to the cytosol and from the cytosol to the nucleus. And transport in, we saw that proteins that are found in the nucleus, like histones, transcription factors, DNA, RNA polymerases, all of those things, they're synthesized in the cytosol on ribosomes, and they have to be imported into the nucleus. And they're imported into the nucleus using the RAN system. Transport out, we have RAN-dependent export, as I showed you, but we also have RAN-independent export, such as the messenger RNA, and I didn't talk about ribosomes, but they don't use RAN either. So messenger RNA and ribosomes are able to be exported out of the nucleus without using the RAN system. So I hope you have enjoyed this discussion of gated transport, and I will see you next time.